right, guys. We got 4 minutes until we get started. Looks like we already have Elliot in here, so welcome, welcome. When you guys join the live lecture, if you could go ahead and put your name down there in that live chat so that I can see that you're here. We will get started in 4 minutes. All right, happy Tuesday. All right, welcome. It looks like we got a few more. Emily, Lance, Laura, Eleanor are here. We got about a minute-ish until 9:40 when we officially start. So just kind of hang out. And I'll say this again here in just a moment. The PowerPoints are posted in our bioenergetics unit folder on Facebook, Schoology. So if you want to follow along while I'm going through everything, feel free to do that. We're going to start with the photosynthesis day 1. So like the overview PowerPoint, that's the one that we're going to talk about today. And we'll have a little bit more to do with that as we get into it. So give it another 30 seconds, a minute or so to get some more people logged on. We got Karina, we got Caitlin. All right, Jack's here. So I'll give it another second to see if we can get some more people here, and then we'll get going. All right. All right, 9:41. Oh, all right, we got everybody. Nice. All right, so happy Tuesday, guys. 9:41, so it's time to go ahead and get started. Thank you for kind of signing on here to YouTube Live and joining me for the live lecture. I did this with first period just a couple of minutes ago, so I have one of these under my belt. My hope is that this one is, seamless, just like the first one. We hit a couple little hiccups there in the first live lecture, but my hope is that I can figure out some of those little difficulties and go from there. So here's kind of the idea with this. I mentioned just before to those of you guys that were on here that the PowerPoint that I'm using is posted in our Schoology materials for bioenergetics. Down at the bottom of the folder, you'll see, I think it's Photosynthesis Day 1. It's basically just the overview for photosynthesis. So that's what we're going to talk about today. There is a second PowerPoint in there that I'm going to ask you to look over for Thursday. So the idea is this. Today is just going to be the lecture. I know that in the weekly agenda I put that there was going to be a live lecture discussion. I decided against that because of just the time. I don't want to have you doing too much during this period. So for this, I really just want you to kind of follow along. Jot down some notes on your end, just like you would do in class. Follow along, whatever it is that you feel best doing. And then before Thursday, I want you to go ahead and look through the next PowerPoint. And that one is titled Electron Flow. So on Thursday, we're going to have a live Teams meeting, and that's basically going to be like our discussion. That's going to be the chance for you to really ask questions, for us to really talk about this information, to make sure that we're good with it before we kind of move on to the next part. Okay, NTI is tough, right? Doing lectures like this over a camera via YouTube Live during NTI is a little bit difficult because I don't have you guys in front of me and I can't see your faces and I can't talk to you kind of one-on-one. But as we use YouTube Live, here's the idea. So I have my computer with the PowerPoint, I got my camera over here, and I also have my iPad set up so I can see the live chat. So the idea is, if something happens with the camera or the sound, or you can't see something, shoot me a little message in the live chat and I'll try to fix it kind of on the fly as we go through it. If you have a question about something that I'm talking about in the lecture, ask that question in the live chat. I'll try my best to watch that kind of as those pop up, and I'll slow down, I'll take a little bit more time, and I'll answer your questions as we go through it. All right? This is basically an overview with a little bit of some more details sprinkled in at the end, but the idea is that we're kind of setting the stage, building the foundation for really starting to dive into photosynthesis this week and next week. Okay, so just follow along with me for this PowerPoint, this lecture. Take notes on your own, follow along, do whatever you got to do. We will meet again on Thursday via a live Teams meeting at the same time, 9:40 on Thursday. That will give you the opportunity to really ask those questions and we can kind of hammer out the information as we go along. Okay, so let's go ahead and dive into it. Photosynthesis, part 1, basically an overview with that. So let's kind of dive into it. All right. So the first couple of slides that you're going to notice in this photosynthesis lecture are basically just examples of organisms that are going to be doing photosynthesis. All right. So we have our autotrophic organisms. So as we think about photosynthesis, one of the most common misconceptions, especially among newer students, freshmen, sophomores, things like that, is that plants do photosynthesis because they have chloroplasts and animals do cellular respiration. All right? What we want to make sure that we understand is the idea that anything with a chloroplast, it can be a plant, algae, phytoplankton, which are actually single-celled organisms, some of which are actually single-celled animals that just have chloroplasts so they can do photosynthesis. Some of them are free-living single-celled plant cells. but they have chloroplasts, so they can do photosynthesis, right? And then we have bacteria. There are some photosynthetic bacteria. Way more bacteria just use other molecules and gases and things like that to get their energy, but there are photosynthetic bacteria that have chloroplasts. They have the structures that allow them to do photosynthesis. So when we think about photosynthesis, we're thinking about these autotrophic organisms, organisms that take in energy from the sun and create their own food. They create sugar, right? Where does that sugar go? The sugar goes, especially for single-celled organisms, algae, plants, that sugar goes to the chloroplast for cellular respiration. Okay, so we have our autotrophic organisms, which have structures that allow them to do photosynthesis so that they can create sugar. On the flip side of that, obviously, we have our heterotrophic organisms, animals, fungi, that are going to need to eat or decompose, break down. If you're a fungus, they're going to need to get that sugar from somewhere else, send it to the mitochondria so that they can create energy. But where does that sugar come from? Plants. All right. So when we think about photosynthesis, when we think about the importance of photosynthesis, right, it's the starting point for everything. Plants, plant-like organisms, things that have chloroplasts, they're going to use energy from the sun with some other things to create sugar, and then that sugar is going to be used not only by them to make energy, but also by pretty much every other organism on the planet that's going to need to get that sugar in some way to get energy. Okay, so just a quick little review of autotrophic versus heterotrophic organisms. Okay, so that leads us into photosynthesis in general. So we're going to start with the chloroplast, and there's not a ton that we need to know about the chloroplast. It's not a class about plants, right? It's not a plant ecology or a botany class, so we don't have to know a ton about the structure of plants or even chloroplasts, all right? What we do want to be aware of, okay, is how can we connect this stuff to things that we've already talked about, okay? So let's look at this really quickly. So we have our plant, we have some leaves, we're going to zoom in on that, okay? As we zoom in on the plant, okay, we're going to see a cross-section, which is going to show some different types of cells, okay? So right here, we have what's called the mesophyll portion of the plant, okay? Within this mesophyll portion, we have these mesophyll cells. These cells are just packed full of chloroplasts. The whole point of a plant having these cells is to be able to do as much photosynthesis as possible with the most number of chloroplasts to achieve that goal, okay? So we have our mesophyll area with these mesophyll cells that are full of chloroplasts, and then we're actually going to zoom in a little bit more on the chloroplast itself. So bear with me really quickly while I zoom in. Obviously, this would be a little bit better in the actual live situation, but I'll try my best to kind of do what I can with this. So when we zoom in on a chloroplast, and I'll show a picture on the next slide too, there is a big idea that we want to pull from this. Okay? Again, we don't have to really know the structure. We don't have to know any of the super specific stuff just yet. But when we look at the chloroplast, there's two things that we want to be aware of. Number one, the chloroplast has an outer membrane. It's a phospholipid bilayer. So just like the cell membrane of a cell, the outer membrane of the chloroplast is made out of phospholipids. Nice little connection between, remember the endosymbiotic theory, where we had these free-living chloroplasts that were engulfed by early prokaryotic cells and led to eukaryotic cells. So it has this outer membrane, okay? What it also has is a super-duper folded inner phospholipid bilayer with proteins that makes up these structures on the inside. Okay, so we have an outer membrane, and then we have all of these kind of sandwiched sacs or pancakes that we call thylakoids that make up the inner membrane of the chloroplast. So what I want you to think about is, yeah, they have the outer membrane, but why would it be important that they have all of this inner membrane? What's the point of that? Why not just have one thylakoid inside the chloroplast, right? So as your brain is kind of cranking, why would we want to have all of this inner membrane inside of the chloroplast? Hopefully your brain automatically goes to surface area to volume ratio, okay? We're going to look at a bunch of chemical reactions that are going to take place on this inner membrane. So the more surface area we have on that inner membrane, the more proteins, right? The more opportunity for those chemical reactions. So the chloroplast can be way more efficient because it has all of this surface area on that inner membrane. Okay? The volume is still low, so we don't have these huge sacs with all of this surface area. They're these nice, tightly packed individual sacs made out of this phospholipid bilayer with proteins embedded in it to increase the surface area. Increase the surface area, more opportunity for chemical reactions, more opportunity for photosynthesis. That's good for the plant. Okay? And plants are going to have thousands and thousands and thousands and thousands of these chloroplasts packed inside of all of these different mesophyll cells so that they can do photosynthesis. Okay? So one of the big things about the structure of the chloroplast is this idea of surface area to volume ratio. What we will also see when we get to cellular respiration is that the mitochondria is pretty similar. It's going to have an outer membrane, but it's also going to have a whole bunch of inner membrane with a big surface area to allow for a whole lot of chemical reactions. Okay, so surface area to volume ratio, I told you early on that that was important, so we're kind of seeing that again here. All right, here's another very simple picture just to kind of give you an idea again of what the chloroplast looks like. We got our outer membrane, all right, in yellow, and then inside we have all of this inner membrane in green. Okay, super high surface area to volume ratio inside with all of that membrane that's going to allow for those reactions. Okay, not a lot other than that we need to know for the chloroplast that we won't talk about as we kind of go through these next parts. All right, so let's look at our overall chemical reaction. I'm going to add something here, right? Okay, so this is going to give us an idea of what we're looking at. So as we go through the process of photosynthesis, the major questions that we are trying to answer are, why is it important that a plant takes in carbon dioxide, takes in water, uses light energy to create sugar, C6H12O6, glucose, oxygen, and then obviously we're going to have a little bit of energy released as heat because we have all of these chemical reactions that are going to be occurring. So we're going to give off a little bit of heat. We don't lose or gain any energy. We're just transforming and transferring it to do what we need to do. All right? So again, the questions are, the question is, how does all of this stuff actually work? Okay? We can look at the overall chemical equation and we can understand what is needed and what is created. But what we ultimately want to do at the end of this is be able to say, this is why it's needed. This is how it's created. Okay? That's what we want to be able to do. That's why we're doing this live lecture. That's why we're going to take the time on Thursday to go over everything and make sure that we're all on the same page. Okay? So, we have our overall chemical equation. We're going to take some time to look at a couple of different processes that are going to help us explain why these molecules are needed, why this stuff is needed, and how that's created. Okay? So, let's dive in. I'm going to zoom out just a little bit so you can see the very top there. Okay, I know it might be a little bit small, but I gave you the PowerPoint so that you can check it out. So the overall process of photosynthesis is broken down into two parts, and here's where we start getting into the really good stuff. Okay? You may remember this from sophomore biology, at least the overall idea. Hopefully that's true. But here are the overall two parts. Okay? The first part is what we call the light-dependent reactions. And I know that, again, that's a little bit small. Hopefully you can see it. So the light-dependent reactions are a series of chemical reactions that start the overall process of photosynthesis by using energy from the sun as well as water to ultimately create ATP, which is right here, and NADPH, which is right here. Okay? So again, big picture idea. The light-dependent reactions, which occur on that inner membrane of the chloroplast, this is happening on those thylakoids. The light-dependent reactions take in energy from the sun as well as water to create ATP, which is our energy molecule, and NADPH, which is an electron carrier, and we'll see more about that here in just a minute. And then it creates this wonderful byproduct that you and I rely on all day, every day, our entire life, oxygen. Okay? So the light-dependent reactions, because they're, they name them the light-dependent reactions, they are going to occur over and over and over again as long as there is a source of light. Okay? When there's no light, we can't do the light-dependent reactions, so we can't create the oxygen, the ATP, and the NADPH. So we need the light to power this set of chemical reactions. Okay? So the first part, light-dependent reactions, it occurs on the membrane, the inner membrane of the chloroplast. Okay? That's part one. Part two, for our overview, is what's called the light-independent reactions, or the Calvin cycle. You may remember that from sophomore year. Okay? So the light-independent reactions, they use the materials ATP and NADPH that were created over here. So the light-independent reactions use ATP and NADPH. They bring in carbon dioxide, and the ultimate goal of the light-independent reactions are to create sugar in the form of glucose, and also to recycle our ATP and our NADPH back so that it can be used again by the light-dependent reactions. Okay? So these two parts, these two major parts of photosynthesis are connected by energy and electron carriers. Okay? Light-dependent reactions, we're going to make two materials. Light-independent reactions, we're going to use that material and recycle it back so that it's ready to work again. Okay? So what plants do, as they're living their day-to-day plant life, they're going to hang out, they're going to take in energy from the sun, they're going to pull up water through their roots, and they're going to use those two things to make ATP and NADPH. Right? As they make these materials, they're then going to be able to turn carbon dioxide using ATP and NADPH into sugar. Okay, so bear with me for just a second. It looks like it's getting a little jumpy. All right, I think we're better there. Okay, so these are the two overall major processes of photosynthesis. Okay, we want to make sure that we understand the big idea, the big picture, before we really start diving in and digging deeper into how these two processes actually work. Okay, so I'm going to give you a second just to kind of digest that. We got the light-dependent reactions. We have the light-independent reactions. Now we're going to start to look at how do these things actually work. Okay, no questions so far on the live chat, so it looks like we're pretty good to move on. So we're going to talk first about this. Okay, so we're going to start with the light-dependent reactions, and the goal is to answer those questions of how are light and water used to make ATP, NADPH, and oxygen. Okay, so we're going to dive in a little bit deeper with that. But we're going to start with the light portion. Okay, you know from sophomore biology or 7th grade biology or physics, we know that the light that comes from the sun, or an incandescent light, whatever it might be, a light source, actually contains the entire visible spectrum of light. Okay? On the shorter wavelength side, which has higher energy, we have our purples and our blues. On the lower wavelength side, or the lower energy side, we have our reds and our oranges. Right smack dab in the middle, we have our greens and our yellows. Okay? So the idea is that plants are going to use the visible spectrum to the best of their ability to gain energy to drive the processes, well, the light-dependent reactions of photosynthesis. Okay? So what does that look like? The idea is this. As we know, chloroplasts absorb all wavelengths of light except for green. Okay? How do we know that kind of right off the bat? When you look at a green plant, it's green. Okay? What's happening? The chloroplasts are absorbing all of the different wavelengths of light except for green. Green is being reflected, it's bouncing off of the chloroplast, it's hitting our eyes, and we see it as green. Okay? Most photosynthetic organisms, most photosynthetic plants on our planet absorb all of the wavelengths of light except for green. Okay? How do we know that other than just looking at them? Okay? There is actually a really cool experiment that I did in college when I took a plant ecology class to kind of back up this idea. We don't have all the tools that we need to do it, but it's actually a really cool experiment to help provide evidence or data to support this idea that plants are going to absorb all wavelengths of light using their chloroplasts except for green. So these next two slides kind of go through that experiment to help us provide a little bit of evidence, a little bit of data for that. All right. So here's the first one, and again, hopefully you can kind of see what we have here. So we have this experiment set up to help provide this evidence. On the left-hand side, way over here, we have our light source. In this case, it's just an incandescent white light. It could be the sun, but it's going to be a light source. We're going to shine that light source through a prism. And if you've ever played with a prism and held it up to a light or the sun, you know that the prism breaks up the visible light spectrum into all of the different wavelengths and colors. All right? Right here, we basically have like a screen or a membrane. And what we can do is we can move this little slit in the screen or the membrane so that only certain wavelengths of light are allowed to pass through. So maybe I'll come over to this side. Okay? So we have our light source, we have our prism, we have our screen. Right here we have a test tube that has a solution with chloroplasts in it. So we have all of these chloroplasts that are suspended in this solution. As the light passes through the screen, it's going to pass through the chloroplasts, and it's going to hit this little tube over here. Okay? What this tube has in it is a sensor that's going to pick up how much light is passing through this test tube into this sensor. Okay? The sensor is then going to allow for a readout on this galvanometer, which basically just picks up how much light is hitting this little sensor. Okay? The more light that hits the sensor, the higher the reading on the galvanometer. All right? So what we can see is that when we shine green light onto chloroplasts, pretty much 100% of that light passes right through the chloroplasts, it's reflected, it's picked up by this meter, and it shows that. Okay? So this provides evidence to support the idea that chloroplasts are not absorbing, they are reflecting green light. Okay? Now, let's change it up a little bit. I know it's a little bit hard to see, but you can see the label down here. So we have the same setup. We got our light, we have our prism, it breaks it up into the visible light spectrum. But in this case, we're going to move our screen, or we're going to set our screen up so that only blue light passes through. Okay? So what the blue light is going to do, the blue light is going to hit our chloroplasts, And then our probe, our tube here, is going to pick up how much of that light is passing through the chloroplast. Well, in this case, it's very little. That means that the chloroplasts are actually absorbing that light energy so that not a lot is passing through. Okay? If we move this opening, right, in this screen, if we move it up to the reds and the oranges and even part of the yellows, or we move it down more into the purples and the really dark blues, we're going to see a very similar readout. Okay? I don't have slides for all of the different colors, but we can actually run this experiment and test the different wavelengths of light and see what happens when that passes through the chloroplast. Okay? And what we're going to see is this. Come on now. We're going to see this spectrum. All right? So what we have here is, down at the bottom, we have the visible light spectrum. We got our purples into our blues. We have our greens and yellows. And then up here we have our oranges and our reds. In this case, we have 3 different pigments, okay, that are going to exist in plants that plants are going to use to absorb energy, light energy from the sun. We have chlorophyll A, which is one of the most common pigments found in photosynthetic plants. We have chlorophyll B, same thing, one of the most common pigments found in photosynthetic plants, right? And then on this graph, we also have carotenoids, okay? Carotenoids are what we call accessory pigments. They're not the main ones, but they are light-absorbing pigments that plants have that they can use for photosynthesis, okay? So what we see with all of these, chlorophyll A, chlorophyll B, and the carotenoids, is that they are in fact absorbing in the purple and blue part of the spectrum. Okay, so all of these pigments are going to absorb the purple and blue portion of the spectrum. Okay, that's energy that can be used for photosynthesis. All of these pigments are going to reflect, so there's very little absorption in the green and yellow spectrum. Okay, and then when we get to this side, we see that only chlorophyll A and chlorophyll B are going to absorb in the red and orange portion of the spectrum. Okay? So chlorophyll A and chlorophyll B, the reason that chlorophyll looks green is because they're absorbing in this part of the spectrum, they're absorbing in this part of the spectrum, but they are reflecting in the green and part of the yellow. Think about the season, the fall season, especially around here, based on the types of plants that we have in our ecosystem, in our biome. All right? During the spring and the summer, and as we start to get into fall, we have these really bright green plants because chlorophyll A and chlorophyll B are showing up in huge amounts. Okay? So photosynthesis is just cranking in those plants. As we move into fall, chlorophyll A and chlorophyll B start to break down, okay? Because there's a need for resources to make and keep those pigments fully functioning that plants don't have as we go into fall. So chlorophyll A and chlorophyll B start to break down, and what starts to take over are these accessory pigments. All right, so what do we start to see? We start to see reflection in the red, orange, yellow, brown portion of the spectrum, okay? So the leaves start to change color because chlorophyll A and chlorophyll B are breaking down, and these accessory pigments are doing all of the work, all right? The amount of energy that's coming from the sun during the fall is much less than in the spring and summer, right, because of the angle of the sun, okay? So there's no need to have chlorophyll A and chlorophyll B working, so plants are going to rely on these accessory pigments, these carotenoids, and that's why we see color changes in the leaves of the plants that we have, okay? They're still trying to do photosynthesis. They're trying to stay alive as long as they can before they go dormant during the end of fall and into winter, all right? So they're trying to do as much photosynthesis, they're trying to absorb as much energy from the sun while kind of saving their resources with those pigments, okay? Why talk about this? because of how important light and the absorption of energy from a light source, the sun, because of how important that is to photosynthesis. Okay? So, how does this work? How do plants actually, using chloroplasts, absorb energy from the sun to drive the light-dependent reactions of photosynthesis? This is where it starts to get really cool. This is where we get to start talking about like the biochemistry. What do plants actually do? Okay? So, plants have these chlorophyll pigment molecules in their chloroplasts. Okay? So I'm going to get you a little bit closer here. I'm going to try to do it so that it's not like super wonky. All right? So here we have our chloroplast. Okay, we have our outer membrane and we have our inner membrane. Okay, on the inner membrane, which is a phospholipid bilayer, we're going to see in the next couple of slides that we have these clusters of pigment molecules. Okay, these clusters are ultimately just light-absorbing centers within the plant. So their main goal is to absorb as much energy from the sun as possible. Okay? Each of these clusters has basically a primary molecule, a primary pigment molecule that's going to have an atom of magnesium sandwiched right in the middle of that pigment molecule. Okay? Why is that important? How are these pigment molecules, how is this atom of magnesium going to actually help this process occur? Okay? So we need to think a little bit about magnesium first. All right? So when we think about the evolutionary history, the way that plants have evolved over time, they've evolved to have these pigment molecules, chlorophyll A, chlorophyll B, carotenoids, all of these different types of pigments. And they've also evolved to have this kind of primary pigment molecule that has this atom of magnesium right smack dab in the middle. Why is that important? Okay? Magnesium, and I'm going to move you closer again. Magnesium has, I know it's a little bit difficult to see, okay? Magnesium has 2 valence electrons, okay? So those valence electrons are going to become incredibly important when we think about the light-dependent reactions, okay? So this atom of magnesium that's sandwiched right smack dab in the middle of this like primary pigment molecule has these 2 valence electrons that are going to play a role in the overall process, okay? So let's kind of take a step back for a second and think about what we've seen so far, all right? So we have the 2 major parts of photosynthesis. We have the light-dependent reactions, where we're taking in water, we're taking in energy from light to ultimately create ATP and NADPH and a byproduct of oxygen. Okay? The materials that are made from the light-dependent reactions are going to be used to drive the Calvin cycle so that carbon dioxide can be brought in to create sugar and also recycle those materials back to the light-dependent reactions. Okay? We started talking about this. How does this work? Well, on that inner membrane of the chloroplast, that's where we're going to have all of these pigment molecules. Okay? Chlorophyll A, chlorophyll B, these carotenoids. They're going to be located in here. And it's their job to absorb that energy from the sun so that we now have that energy to drive these reactions, right? How cool is that? Okay? So, let's look at how this process works. Okay, so we have these things on the inner membrane of the chloroplast that are called photosystems. Okay? Photosystems are just specialized proteins whose main job is to have these pigment molecules available to absorb energy from the sun, from a light source. Okay? So all of these, and I know that it's a little bit difficult to see on this screen. Maybe you can see it a little bit better if you're following along with me. But what you can see is these right here in this image are a lighter green color. And then this one right here, which we call our reaction center or our primary chlorophyll pigment molecule, is dark green. Okay? So all of these molecules around the outside, their job is to absorb as much energy as possible. Okay? As they absorb that energy, those electrons are going to become excited, and they're basically going to move from molecule to molecule until those electrons get to this kind of reaction center, this primary chlorophyll molecule. Okay? When those electrons get there, we're going to have some cool stuff happen. Okay? We're going to have a series of chemical reactions that are going to allow us to use the energy from the sun to create ATP, NADPH, and oxygen. Okay? So hopefully we're good up to this point. I don't have any questions showing up in the live chat just yet. I'm trying not to go too super fast while still kind of building these layers of how this process is going to work. Okay? So we're here now. Inner membrane of the chloroplast. We're on this protein called a photosystem. We have all of these chlorophyll pigment molecules that are actively absorbing energy from the sun to excite electrons. Okay? And again, like I said before, this is where it really, really starts to get cool. Okay? So I'm going to go to this next slide. So this is what happens. Okay? So follow along with me because I know that there's a lot going on this slide, but I want you guys to try your best to follow along with me as we kind of look to see what this looks like. Okay, actually, let me show you this slide really quickly. So this is where we are. Okay, so I'll zoom in a little bit here so that you can see it. Okay, so we have our chloroplast up here. Okay, we have our outer membrane and we have our inner membrane. We're zooming in on one portion of the inner membrane. So what you can imagine is that we have tons of these proteins all over this inner membrane. Okay? It's these proteins that are, some of which are embedded in the membrane. So we have integral protein, integral, 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 integral. Here we have some peripheral proteins that are going to work in the process too. Okay? So it's these proteins that are going to work together to use the energy from the sun to ultimately create ATP, NADPH, and oxygen. Okay? So I'm going to go back one slide and I'm going to show you guys how this works. Okay, again, if you have any questions, if you're not sure exactly what's going on, go ahead and throw them down in the live chat. Okay, so bear with me. Okay, so here's how this works, and I'm going to zoom in over here so that you guys can see this. Okay, so here's how it's going to work. This whole process starts at what's called photosystem 2. Okay, we also over here have photosystem 1. The question is always, why is photosystem 2 first and photosystem 1 is second? Well, it turns out when they were doing the research to discover how this process worked, this was the first photosystem that they discovered. This was the second photosystem that they discovered. And as they figured out more about this process, they figured out that this one actually came first in the process. They didn't want to change the names at that point. So photosystem 1 here, photosystem 2 there. Okay? So follow along with me. Check out how this works. Okay? So on photosystem 2, we have all of these pigment molecules that are going to absorb energy from the sun. Okay? As those electrons are excited, they're going to work their way to this kind of primary pigment molecule, which we call P680. You don't need to know that. Okay? Those electrons are, at this point, they are so excited that they're going to jump from their outer electron shell and they're going to move to this first protein, which is called a primary electron acceptor. Okay? Again, sorry, it's kind of jumpy. The primary electron acceptor is just a step in the process of moving these electrons down a series of membrane proteins so that we can get to our ultimate goal of making ATP and NADPH. Okay? So the two electrons are going to jump onto the primary electron acceptor. They're going to then move to what's called PQ, which is plastoquinone. You don't need to know the name of that, but that's just an in-between. And then they're going to move to another membrane protein, which is called the cytochrome complex. Okay? The cytochrome complex is going to, it's actually a proton pump, an H plus pump. That's going to pump hydrogens to create ATP. So this is where we make our ATP. So we're going to use some of the energy from those electrons to make ATP. Those electrons have lost some of their energy, so they're going to move to plastocyanin and then to photosystem 1. Why do they have to go back to photosystem 1? So they can be re-energized. As those electrons are re-energized, they're going to jump to another primary electron acceptor. They're going to go to ferrodoxin, which you don't have to know, which is just an in-between protein. And then they're going to wind up right over here at this protein called NADP plus reductase. What is the point of that protein? That is to take NADP plus and 2 hydrogens. It's going to add in those 2 electrons and create NADPH, which is an electron carrier. Okay? So the electrons that started over here, that were energized by the sun, are going to move through a series of membrane proteins back to photosystem 1 so they can be re-energized. Then they're going to move through another series of membrane, They're going to move through another series of membrane proteins until they wind up in this molecule called NADPH. Okay? NADPH is ultimately going to carry those electrons to the light-independent reactions or the Calvin cycle so that that process, that part of photosynthesis can work. Okay? Where does the water come in? Because if you remember, and I'm going to bring you up here really quickly, if you remember, the light-dependent reactions take in light and water in order to make NADPH, ATP, and oxygen. Right? So we saw how the light plays a role in making the ATP and the NADPH, but what we need to look at now is where does the water come in? Okay? So I'm going to bring you right back down here. Okay? So as these electrons are excited, they're going to physically leave and wind up in NADPH, which is over here. So we have to have a way to replenish the source of electrons so that we can keep making NADPH. Those electrons come from water. So one of the reasons that plants need to take in water is so that they can constantly break down these molecules of water and replenish those electrons. So every time 2 electrons leave, the plant breaks down a molecule of water and replenishes those electrons so that they can leave again. All right? So something that is really, really cool to think about is this. Okay, so bear with me for a second. Something that is really super cool to think about is that the oxygen that we breathe comes from water. Plants break down molecules of water to replenish those electrons, and because they break down that molecule of water, they give off oxygen. That oxygen diffuses out of the plant, goes into the atmosphere, and we breathe it in. Okay? So when you think about northern Kentucky and greater Cincinnati, right, we have the Ohio River. As that water, right, evaporates from the Ohio River and condenses into clouds and rains down to all of our plants, and all of those plants do photosynthesis, they break down the water that ultimately comes from the Ohio River, and they create oxygen. So you're breathing, as you sit in your house or wherever you are, you're basically breathing Ohio River oxygen, which I think is kind of cool. Okay? So with our light-dependent reactions of photosynthesis, okay, we have our light right here, energizing the electrons. Light energizing the electrons. So that's why plants need to take in light energy, okay? That light energy excites those electrons so that we can make ATP and we can make NADPH, and those can go to the Calvin cycle. Plants also need water so that they can break down those molecules of water to replenish the electrons so that the process doesn't shut down. Okay? If you don't water your plants, they can't do photosynthesis because they don't have the electrons that they need to make NADPH and ATP. Okay? That's the light-dependent reactions of photosynthesis. Okay, so I'm going to go to this last slide here, and I'm going to show you kind of what it actually looks like if we embed these proteins in the inner membrane of the chloroplast. Okay, so again, I know it's not perfect, but this is basically another view of what we saw on that last slide. Okay, here's photosystem 2. This is an integral protein in the phospholipid bilayer. Okay. Here's what we call the cytochrome complex. This is just a proton pump. All right, so the whole reason that this is in there is to pump those protons. Here's photosystem 1, which is an integral protein. Here's NADP plus reductase, which is also an integral protein. Along the way, we have PQ, PC, and FD. These are peripheral proteins. Okay, so they're going to work to help the process of moving those electrons. Over here, we have our NADPH, and you can see that that's going to go to the Calvin cycle. All right, and down here, we have our last integral protein, which is called ATP synthase. And ATP synthase is going to create ATP every time that a hydrogen is pumped through to make that ATP. Okay? So as we zoom out, and we think about this inner membrane of the chloroplast, we have this series of membrane proteins that are going to work together with light energy and with water to create the materials that we need to drive the light-independent reactions or the Calvin cycle, right? We have our NADPH, we have our ATP, we're splitting our molecules of water to create our oxygen and to replenish those electrons that we need. And all of these reactions, right, each stage is a chemical reaction. So we have chemical reactions occurring here as those electrons are moving. We have chemical reactions that are occurring here, right, as these proteins are changing shape and moving the electrons down the line. We have chemical reactions occurring here and here and here. So it's this series of chemical reactions that are all going to work together to create the materials that we need for the light-dependent reactions. Okay? This is the awesome stuff. This is the stuff that we do not talk about sophomore year because it gets into a lot of the actual biochemistry, the science behind how this actually works. Okay? Should be our last slide. So, to review, and then we're going to wrap it up. We got about 20 minutes until the official end of the period. Oops. Trying to go back, but it's not letting me. See if I can zoom out here a little bit. Okay. So, to review, and then I'll let you guys go. We have our two major parts of photosynthesis. Today we talked about this, okay, at least the starting portion of this. How on the inner membrane of the chloroplast, on those thylakoids, we're using energy from light and water to create ATP, NADPH, and oxygen. The main goal is that this ATP and NADPH are going to later be used to power the Calvin cycle, or the light-independent reactions. Bring in carbon dioxide, make glucose, and recycle those materials back to be used later, okay? The water is going to be split to create the oxygen, which is a byproduct which is released from the plants, and we breathe it in, okay? So what I want you guys to do now, okay, we got 20 minutes before the end of the period. We're going to meet again on Thursday via Microsoft Teams, same time, 9:40, right? What we're going to do during that live meeting on Thursday is we're going to talk through this stuff. So I'm going to give you guys the opportunity to ask questions. We're going to dive a little bit deeper into this and look at some more information about the light-dependent reactions, so that we can really kind of create this picture of how this process is working, why it's important, and how it's ultimately going to lead to the light-independent reactions, or the Calvin cycle. Okay? I'm not going to go any further today. Thank you for joining in. Thank you for putting your names there in the live chat, so that I can see that you're here. All that I want you to do again, kind of digest the stuff that we talked about today, look through that second PowerPoint, and come up with some questions for Thursday. I'm going to really let you guys drive the live meeting on Thursday. You're going to throw some questions out there. I'll share my screen, and we'll talk through it and see if we can't make some sense of what's going on with the light-dependent reactions. Okay? So that's all that I have for today. Enjoy the rest of your day. We don't have anything tomorrow because it's an even day, but we'll come back together on Thursday at 9:40 for a live Microsoft Teams meeting and go from there, okay? I miss you guys. I wish more than anything that you were here in the room with me so that we could really talk through this, but we're on NTI, so we're going to try to make the best of it. I will see you guys on Thursday. Enjoy the rest of your day. All right? Take care.